Hello, Assalamu alaikum students. Welcome to MBBS Online Lecture 2021. Topic for today's lecture is about the ABO blood grouping and typing. So let's discuss it without wastage of time. First of all, what are the importance of the blood groups? Why it is done? And these are the learning objectives. So discovery of blood typing. Who discovered blood typing? That is a scientist named Dr. Carl Landsteiner. He also gave the law of Landsteiner law that we would discuss in further in the lecture. Okay. He determined that blood typing in a human also Rh positive and Rh negative, and it is because of this scientist that many transfusion reactions are not happening in today. So blood typing, determination of blood groups of recipient and donor on the basis of presence of or absence of antigen that are agglutinogen in RBC on the surface of RBC. That determines the blood type. Means what are the antigen present on the surface of RBC, that these determine the blood typing. Importance, blood typing is important for blood transfusion, mainly blood transfusion. Certain blood disease, paternity test, forensic medicine. Paternity test, how it is done? If a father blood is A, so his male child blood would be also A or A positive. For example, A positive of the father, so A would be probably blood group of the child would be A positive. So this is also called paternity test. Blood transfusion reactions mean if a mismatched blood is transfused in the recipient body, and that may cause hemolysis of the blood and causes a person to be anemic, severely anemic, and may cause multi-organ failure in that person. So therefore, blood typing is necessary before the initiation of blood transfusion. Okay, forensic medicines. In forensic medicine, that is about fingerprint testing or DNA testing and all of that stuff. Okay, so let's do the next slide. That is about different blood groups. What are the different types of blood group present in the population? First, number one is OAB blood groups. Then number two is RH system. Number three is MN system, and other are SS, small s, P, DA, or T, and these are kind of stuff. So the most important are number one and number two. That is OAB blood groups in RH system. Because these two blood groups are mainly concerned with the blood transfusion reaction that occurs while due to the mismatched blood transfusion. Antigens and antibodies in human blood cells. What are antigens and antibodies in human blood cells? In RBCs. So, agglutination. What are agglutination? That are basically antigens. That are antigens present on the surface of RBC. These determine the blood group of that person. If there are two types of agglutinogen, that are agglutinogen A and agglutinogen B. If person having agglutinogen A, his blood group most probably would be blood group A. If agglutinogen B is present on that person's surface of RBC, so person blood would be agglutinogen B. So these are basically protein compounds, compound complexes that are present on the surface that when combining with the corresponding antibodies that are complementary to the shape of antigen and cause agglutination. Okay. Let's discuss next slide. That is about agglutinogens. For example, group A, there are four types of blood groups: group A, group B, group AB, and group O. Group O having no antigen on the surface. No antigen are present on the surface. While group AB has both antigen present on its surface. That is A and B. While group B having only B antigen present on the surface. While group A having only A antigen present on the surface. These are also called agglutinogens. Antigens are also called agglutinogens. These antigens are called agglutinogens because they often cause blood cell agglutination, cause blood transfusion reactions. So, what antigens are involving in the blood transfusion reactions? That are agglutinogens that may combine with the corresponding antibodies and may and cause clumping of RBCs. We would discuss it later. Okay, genetic determination of agglutinogen. If a person having blood genotype OO that are zero, this is also called zero. If both the homologous chromosome has no allele for the blood group, so person blood would be type O that produces no agglutinogen with no antigen on the RBC surface. While genotype OA or AA means OA means one antigen A on the one chromosome, while no allele on the second chromosome. That process produces agglutinogen A, while group genotype OB and BB produce agglutinogen B, and genotype AB that produces agglutinogens A and B. Both are present on the surface of RBC. Agglutinin. What are agglutinin? These are basically antibodies, which are immunoglobulins IgM or IgG that cause agglutination on combining with corresponding anti-agglutinogens are called agglutinins. These agglutinins are the antibodies that, when combined with the corresponding complementary antigen on the surface of RBCs, may cause agglutination of the RBCs, that is, clumping of RBCs to each other, adherence of RBCs to each other, and may occlude small arteries or veins. Types: there are two types, anti-A agglutinin and anti-B agglutinin. So these are also immunoglobulins of IgM or IgG type that are all present, mostly present in the plasma. We will discuss how these antibodies occur in a person plasma and how these develop. So agglutinin, so what we have discussed agglutinins that are antibodies. Agglutinins are gamma globulins in plasma. When type A agglutinogen is not present in person's blood, antibodies known as anti-A agglutinin develop in plasma. This is also a Lenz-Steiner law that that states that if person antigen A is present in person's blood, so its corresponding antibody or agglutin would be absent in that person's plasma. And vice versa is for the type B. If type B antigen is present in the person's plasma, so antibodies B would be absent in that plasma. And instead of, for example, in type A, if type A antigen is present, instead of type antibody A, antibody B would develop in that person. So I will discuss it again, just like when antigen A is present in the person's blood, so its corresponding antibody would be absent. But its opposite antibody would be present, just like if antigen A is present, anti-A agglutinin would be absent, Type B is agglutinin. The four classic types fill in the blanks. Substance that can trigger the production of antibodies that are basically are the antigens. Antigens are the foreign substance that can, when enter in a person's blood, can trigger immune response. While in the second one is antibodies. APO blood group. Two genes, one on each of two paired chromosome, homologous chromosome, determine the APO blood groups. As we have discussed with genotype and genetics. Type O is either functionless or almost functionless, so that it causes no significant type O agglutinogen on the cells. Means no antigen. If type O is present, gene is present on both the chromosomes, no antigen would be present on the surface of the RBC, means zero antigen. That's why it is also called type 0. Type A and type B genes to cause strong agglutination on the cells, means strong agglutinogen means presence of antigens corresponding. So this table is very important. So just like we would understand it better in this table, for group A, its antibodies in plasma would be anti-B, not anti-A. And antigens in the red blood cell would be A. Therefore, the blood group is A. While for the blood group B has antibodies in plasma that are anti-A, antibodies A. No antibody B would be present in the person with the blood group B, and antigen would be B. While for AB, both the antigen would be present, but no antibodies would be present in the plasma. That's why, because both the antigen are present in the plasma. While for blood group O, no antigen are on the surface of the RBC, so they may cause antibodies, both the antibodies present in the plasma. How agglutinin antibodies develop in the plasma? This is because when a fetus is born and he or her having a blood group A, for example A, so no antibodies would be present in that person's blood, not anti-B or not anti-A. But when an anti-B, when antigen B, blood group antigen B, enters in its body through food, through bacteria, or other ways, that may trigger an immune response that may cause the production of IgM or IgG antibodies that are also called anti-B agglutinins. This is how the anti-agglutinins or antibodies develop in the plasma. These are not present from the first day. These develop when we are exposed to some antigens, to foreign antigens. Okay, there's a Lenz-Steiner law that I have produced at the start. It states that if an agglutinogen is present in RBC, its corresponding agglutinin must be absent from the plasma. But its opposite agglutinin would be present, such as if a person has agglutinin A, means blood group A, he cannot have agglutinin anti-A, but he may have anti-B. This is the Lenz-Steiner law. 4 OAB blood groups are most common in the human population. That is blood group O, blood group A, blood group B, blood group AB. We have also discussed the genotype of these four blood groups previously. That is, if O is present, blood group would be O. If OA or AA would be present, blood group would be A. If OB or BB is present, blood group would be B. If both the AB genes are present, so blood group would be AB. Universal recipient. What is the concept of universal recipient? That is, universal recipient is a person with AB blood groups. Means if a person having AB blood group, so he is termed as universal recipient. Because in AB blood group, no Agglutinins are present in the person's blood. So when a foreign antigen enters in the person's blood, it may not initiate immune response. So no agglutination occurs, so no hemolysis of the RBCs occurs. That's why the person can receive blood from any of the type, any of the blood type. As I have discussed, it AB, no antibodies are present in the plasma. So they may not cause agglutination. Okay, so always just like this is genotype and all of this table is discussed in this table. No antiglutinogens, sorry, agglutinogenic are present in blood group O, so both the antigen are present. Same is case for the blood group A. Okay, blood types. To whom person can donate blood to, into whom person can receive blood from. This is all discussed in this table. So blood group A have antigen A while antibodies anti-B. He can donate, he or her can donate to he or she can donate to A or AB, a person with blood group A or AB, and can receive from person with A or O blood group. And this is all discussed with this table in this table. It is also with the genotypes. What are the alleles present on the surface? Oh, sorry, on the chromosomes. Okay, distribution of blood types within a population. Here we can see the most abundant blood type is 46%, that is blood group O, while second number is blood group A, that is 42%, and B blood group is 9%, while AB is the most rare, that is 3%. This is also Rh positive and Rh negative. Okay, Rh positive would be 85% in the population, while Rh negative is 15%. And we can match with just like O positive and O. Okay, universal donor. What is universal donor? Person with blood group O are called universal donor. Means it can donate blood to any of the blood types. It can donate blood to any of the person with any of the blood type, because this will have neither A nor B agent. Means it cannot trigger an immune response in the recipient's blood. So agglutination and hemolysis and transfusion reaction. How this is occur? When a mismatched transfusion reaction or transfusion process occurs from a donor to the recipient, it may cause hemolysis or agglutination. So when blood are mismatched, so anti-A or anti-B plasma and agglutinin are mixed with the red blood cell that contain A or B agglutinogen respectively. Means anti-A, so anti-A antibody comes in contact with its corresponding antigen, that is A. They may that may combine with the antigen and antibody. These both combine and form antigen antibody complex. This complex can result in agglutination, that is clumping of RBCs that may occlude small vessels, or it may induce direct effect of antibodies or indirect effect of antibodies. Direct effect is opsonization, that is coating of the antigen by antibodies that may enhances phagocytic actions, neutralization of the toxin, or precipitation, while indirect would be complement initiation of complement system, that is the complex of 20 protein that works in a cascade manner and may result in the formation of membrane attack complex that may cause hemolysis of the red blood cell, which is called hemolysis. This is also discussed in this slide. This may cause releasing of hemoglobin into the plasma. Hemoglobinemia could occur. So this is the most fatal fatal condition we can say, that is the most dangerous, involving blood transfusions. Direct effect I have discussed. Now agglutination. During severe hemolytic reaction, fever chills. Okay. Blood typing. How blood typing is performed in a laboratory. Let's discuss in this picture. That is most important. For example, if anti-B is present, if for example if type A is there, and we have exposed it with anti-B antibodies that may not clump with each other, that may not cause hemolytic action. So the blood would be liquid. While in if anti-A is treated with A type A is treated with anti-A antibodies, the clumps would be formed. We can see in the picture. Same case is with type B. If anti-B is treated with type B, clumps would be formed. But if type B blood group is treated with anti-A antibodies, no clumps would be formed. And same as the AB. The blood principles of blood transfusion. What is done to prevent blood transfusion? There is blood typing, transfusion, the same type of blood, cross matching. Okay, that's all for now. Thank you for watching this video. Hope you like it. Share and subscribe it with your friends. And we will see you in the next.